So, okay, good. So let's just start. So, of course, first, again, I want to thank you, Fairly, and also Axel, for organizing this such a fantastic workshop. I think it's very helpful. I hope I attended that maybe, for example, 2 years ago, so that I may get, more instructions or something like at least get more experience according to that direction. So first, a brief introduction for myself. My name is Peijie Zhou, and in the math department, and my mentor is Professor Jing Nie. Maybe some of you already know him. So, previously my PhD work was not about single cell analysis or has little to do with the data. Actually, I'm just doing some more relevant to system biology, like some equations, like circuit, to build that, or some more mathematical analysis, formal analysis of the equations. But after I came here, I just actually I think our whole group switched the direction to the single cell data analysis, and I just tried something and did some project, and actually I think I won't say got some experience, because there are some good things or bad things about it. I would just want to share, my feelings about or my personal point of view about some topics. So of course today, actually told me to talk about the dimensionality reduction. Of course, that is a very important step in the single cell analysis. So I would try to combine, different angles, maybe some kind of from mathematics, but trust me, not so theoretical, some kind of from practical, about some, practical cautions. So of course, I know there will be a very good notebook about how to do that using the pipeline. So actually this talk, this lecture is actually intended for just a more in general introduction about this topic, and some just like some high level things, but that I think might be a little bit value. So yes, let's just start. Yeah, okay. I will use this one. It's okay. Oh, I need to mute this one. This one.
So mute that one and then
Okay.
unmute this one and then see.
Let me see. Okay.
There you go. Okay, great. Thank you.
Thank you. So yes, I'm not sure whether we have already talked about other pipelines or. Okay, this is the first time, right? For this.
We talked about mapping reason.
Yes. Oh, okay. Yes. So that's exactly the first step. Sorry, that is exactly the first step, right? You have the sequencing, and then you map a count matrix. It's actually a matrix, for example, just like many genes times many cells. So of course, then you do some quality control, like for example, some genes not expressed, or some cells are low quality. And anyway, maybe there are also some normalization tools, or some pipelines to select some highly variable genes, for example. So anyway, before the formal analysis of this dimensionality reduction, is actually the main goal, mathematically speaking, is just like we have a vector for each cell. There's a vector of RP, and actually P, for example, you're using Seurat or anything popular pipelines, you select 2,000 genes that is assumed to be highly variable. They're more informative to show the heterogeneity of this whole cell population. And our goal is just like to anyway change that by using some mapping or functions, whatever you have, to map this RP to RK, and K is very small. So typically it depends on your goal. For example, you're doing visualization. So here, visualization is important, provide some intuition. So actually for our human eye, we can only understand things in 2 or 3 dimensions, right? That is most for visualization. But of course, actually for dimensionality reduction, we talk about, in a more, in a broader setup. Actually, K can be something like, for example, 20, 50. That is especially when you do some efficient or very simple dimension reduction, like for example, PCA. Maybe many of you have heard about that. So actually keep 50 or 100 PC, then you do the rest. So actually dimension reduction, anyway, the mathematical goal, I think, is very clear. The ultimate goal is you make a, or we call it projecting from a high dimensional space to a low dimensional space. So actually that's a very good point. You can see from this pipeline, so actually dimension reduction is in the circle. It's actually, connecting with the past and also connecting with the future, because actually from visualization have understand about the data. That's one thing. But also, for example, you have, later we talk about, you have different PCs. Actually, based on this PC, you can do the rest. For example, do the clustering, many of the downstream analysis, for example, a clustering or actually, for example, inference, many of things actually rely on that, rely on dimension reduction results. So why dimensionality reduction is a very key step in my mind in the whole workflow of the single cell analysis. So of course, first of all, let's just still want to provide some basic motivations for why we need to do dimensionality reduction. So first of course, if we just want a short answer, that is called something called curse of dimensionality, right? We don't like high dimensional data. For example, the common dimensionality is just like, as we said, after you do the alignment, you have a cell by gene matrix. So by default, maybe just like 20,000 genes, but actually, maybe this data itself is actually quite sparse, not many genes expressed, or actually for the process you want to concentrate on, not so many genes, not so many pathways are involved. So that's why we want to, some kind of to get more condensed information about that. So of course, there's also some theoretical limitations. For example, mathematically, I won't touch that too deep, but something just like the high dimensional data has some property that is not so well posed. And also, even from a practical perspective, from the computational cost, right? You have large vector, you have large metrics, either store that or you run the different algorithms. So that's very hard. But for a very small vector, for example, from the 2000, you just reduce that to, for example, 10 dimensions. Of course, that is better, save some space and also save computational cost. So that's another thing. Anyway, that is a very short answer, curse of dimensionality. High dimension, some kind of, high dimension is something that we don't like, it's undesirable. We like low dimensions. And also, as we said, the sparsity of the data, right? Maybe many of you also know that single cell data, its unique feature is actually sparse, many zeros in that. And also this can be technical, but biological reasons. And also what I mean biological is that, for example, we said actually the informative genes, the important genes, they're just in a few pathways that are important in the whole process. So actually we don't want them as whole. And also, something about noise filtering. This is also one thing, some of the single cell sequencing, actually, from the platforms, from technology that's not so accurate for the different kinds of counts. So actually maybe noise into that. So actually a successful, dimension reduction can also handle with noise. And also I think another topic, maybe we want to talk about later throughout this workshop, something called batch correction or batch information, is that you have different platforms, different labs, different tissues, want to integrate them together. So actually, if you just do it in an original space, it's very hard, but you do something called latent space. In the low dimensional case, actually focus on the key pathways or the key gene sets or the key dimensions, actually that's easier. That is also the basis for the many currently mainstream batch correction or data integration algorithms. Even for multimodal data stuff, for example, we have RNA-seq, we also have ATAC-seq. That's also the basis. We first do something that is not so uniform because in the original form, but actually after you do something, you put them in the latent space, we can assume, okay, latent space, something is more more general, more universal, more unified, so we can have different treatment together. And then of course that thing is just like it's very important to understand the data for the human being, right? From the human eye, we just want to see what the data looks like, so that's for visualization. That's also important for a high dimensional, for example, matrix. How can we reduce to the scatter plot on the plan? That is also very important motivation. So I think that's why we need dimension reduction in the single cell analysis. And for the different categories of dimension reduction, I think there are many ways to classify that. I think this is possible one approach from the methodology perspective. Just recall, just the math goes very clear. We have a large high dimensional vector we're going to project into low dimensional space. So depending on the projection function, the form, we have some linear mapping, that's PCA or ICA. We also have something called nonlinear mapping. That's also maybe some fancy name called manifold learning or something. But actually the algorithm is also very popular, like t-SNE and UMAP. Many of you heard of that. And also there are some thing called graph-based. For example, they're actually more relevant to the lineage for the downstream analysis like PAGA or like spring or false atlas layout that we also going to talk about today. And the thing we won't mention, but that's definitely a very, very good topic. Maybe I would suggest we can also maybe talk about later through this quarter, through the series, is about deep learning methods, how to do that. So the first way, of course, we just today focus on the traditional methods instead of the deep learning methods, but actually that's very important. Like CVI, CVI is actually, or the variational autoencoder, actually also some fancy, but also very useful ways for the dimension reduction. So first, of course, I think I will mainly focus on PCA, because I think of other methods, maybe many of them very fancy, they publish in very high impact journals, but let's just, in my mind, let's just go back to original. I think always the most important one is the most classical one is the PCA, although it's, for example, is proposed 100 years ago, but still is the most important thing to understand, to have a great understand about the dimensionality reduction. So why PCA is short for principal component analysis. So why it is important? First, it's simple, but it's also efficient. It's very easy to calculate, but it's so actually that is the first thing. You run a standard pipeline, you want to calculate the PCA. The second thing that is also very important in the current single cell analysis, you can interpret the results, because both mathematically, literally provide some mathematical understanding, but you know, biological, that's also important, because actually the principal components, the PCs, they are actually in the original gene space. You can link the results with our original genes, but that is not the case for others. For example, you map t-SNE, you can only do that. Based on that, you do some downstream analysis and recover the genes, but not direct connection with the original gene space and the reduced coordinates. That's also important, you know, that is interpretable. And also that is as we said, is the basis for downstream analysis. For example, using t-SNE, for example, when you do the t-SNE, actually the best practice is not directly use the original metrics to do the t-SNE, but rather pick, for example, the top 50 PC and then do the t-SNE, or UMAP, or construct the KN, or do the clustering. So actually PCC is the first step of all the downstream analysis. So actually PCA is beyond merely visualization. So actually maybe PCA is not the best way to visualize. That's a limitation of PCA. That it's not so powerful because a little bit too simple, but actually this is a very important dimensionality task, because our ultimate goal is not only about visualization, about the data set. So here we just briefly talk about some interpretation, more mathematical, but I hope that it can be better to understand what is the basic logic PCA and to use PCA, you know, more wisely in the application. So I will provide 3 interpretations of how to understand PCA. So first, understand it like that. For example, each point here represent one cell, and they're, for example, they can also in the high dimensional space. So what is PCA, or what is principal component? The first interpretation is that is a direction. So principal components, just like direction, for example, is a vector. We have 2000 dimension space. PC is actually one, for the first PC is still 2000 dimension vector. That is a direction, right? Direction in a high dimension space. But the first PC is the direction where the data has the largest variance. So variance is actually a good thing when you do single cell analysis. You want to tell, okay, which direction separate the parts, separate different types of the cells. So that's very important. So the first PC is the direction that has the largest, and the second PC, you can understand that direction that the data has the second largest variance. So actually PC, other directions where the data varies most, or means it's more informative about the current analysis. And after you find this, so the basic pipeline of PCA is just like first they identify these important directions, and given these directions, like for example, they choose the first 10 PC, what they do for each cell, what they do is actually the cell is still in the 2000 dimension space. You pick one PC, you just make it, for example, inner product, or that is projection. So that for each cell, now you have 10 number. If you pick up 10 PC, if you have 20 PC, you do projection on the 20 directions, you have 20 number. So that's natural way for the dimension reduction. It just means, okay, so how similar this cell is relevant to that PC, so what is the relative weight to that direction? So that's the basic first understanding about what is principal component analysis. So anyway, the output, you give a very large, long matrix. The output just depends how many PC you want to truncate. That is the output is just like it's called the PC scores on the different directions. The first interpretation. So second is quite relevant, maybe more abstract, but actually this is something tell you, okay, the different PCs, different directions. If you know a little bit about linear algebra, they span linear space. So actually PCs is actually spans the linear space that best approximates the original data. So here I just mean the linear space. It's just, of course, you can do nonlinear ways, but actually, if we only do the linear way. Actually, there's a theorem to prove PCA is the best approach. There's no other approach is better, can beat PCA if that is the linear transformation to the data. The linear transformation, it means just, okay, each one is just like linear combination of the coordinates to make a new coordinates in the low dimensional space. So actually, it also means, PCA creates a new coordinate system because it's like the PCA actually, as we said, they span linear space, or actually they are orthogonal basis. So actually they create a new coordinate system that you can project. So the third interpretation, that is also interpretation that we are going to talk later, is something called preserving the distance. Because, another main goal, whatever method that is to do the dimensionality analysis, dimensionality reduction, is to preserve something, preserve the information in the original space into the reduced space. So actually PCA can be understand a thing called preserving Euclidean distance. So for example, you have the data points in the high dimensional space, you have something called Dij and Xi and Ij are the coordinates you want to produce. If you want to do that, actually you can directly prove that is the best approach. It's also PCA if you want to preserve Euclidean distance. So after do the PCA, the distance in the low dimensional space can best preserve that in the high dimensional space. So that's another understanding of the PCA. So actually distance, what does distance mean? It's just like biological similarity, right? The Euclidean distance is an understanding. You understand two cells as two vectors. So if that distance is very large, the cell are dissimilar, but if they are similar, means the distance is very small. So this is also very important. So after do the PCA in the reduced dimension, you find some closed cells, means maybe in the original space, they are more biological similar. So that's another interpretation. But of course, we talk about so many good things about PCA, but there are also some bad things or limitations of why we need more, more advanced methods. So first, of course, limitation of PCA is like it's actually good for dimensionality reduction, but that's not good for visualization. So currently for visualization we use t-SNE UMAP. So but actually that is star is one thing is something called overcrowding. It's actually they tend to some kind of make the points too close in the pieces if you only plot two, because by visualization I mean we only plot the first two components or at most of three. That's called visualization. But actually, maybe PCA is very good if you look at 10, 15, but you can not easily to represent that. So that is the issue. It's not so powerful, or the first two PCA are not so condensed for the information, but look 10, that's totally okay. Second is something about inconsistent assumption is PCA is a linear transformation. Assume the data is something like linear, but actually, mathematically speaking, something is called low dimensional but nonlinear manifold. You can imagine the data itself is low dimensional, but it's something like it's like a surface. It's actually, for example, it lives in a three-dimensional space, but its intrinsic dimension is 2, but actually something is folded. But PCA cannot do that very well. It's not a linear transformation if you want to open that row, for example. So that's why something called existent. So something nonlinear, I would just make another comment because actually nonlinear is actually very common in biology, right? Maybe the nonlinear assumption is the better assumption in biology because, for example, if you know something about how to model gene expression, actually the expression genes use some Q functions or some regulations to control. That's intrinsic nonlinear. So that's why it's better to use nonlinear methods to analyze single cell data. You can assume, okay, they are generated from nonlinear process. So that's why maybe nonlinear methods can be better. So now we just talk about something to t-SNE, something called t-distributed stochastic neighbor embedding. So actually what t-SNE do, first let's go. Yes. Yes, I think it just depends on the different, different choice. You can normalize it. You can either center it or you can make that, the variance, the standard deviation is 1. So actually it's something this more empirical, I will say. There's no so much theoretical guarantee about that, but yes. Yes, of course, that is important. Yes. Yes, Gaussian, or for example, take log transform or something. Yes, of course. Yeah. What does mean by mutual connection? Yes, but I think that more relevant to the nonlinear methods. But entropy is not something linear because Euclidean distance, that's something linear. But yes, thank you. So now we talk about t-SNE. So actually, when you do the t-SNE, the basic, remember in single cell, the basic workflow is that you first get 20 PC. You first do a PC. For this 20 vector, you want to further reduce that in the 2-dimensional space. That is the popular approach. Although you can also directly from 2000 to 2, but actually currently people suggest it's better to use 20 PC or 50 PC than to use the t-SNE to further project the data into 2-dimensional space. But this, the first projection is linear in the PCA, but the second projection in t-SNE is something non-linear. So another way, I won't talk too much about mathematics, but a way to understand t-SNE, you can understand from PCA is preserve Euclidean distance, but t-SNE you can understand is preserve some nonlinear Gaussian similarity. So what does it do is actually X are in the high-dimensional space. You somehow define some complicated nonlinear data similarity. For example, larger data, you think that they are more similar, but this is no longer linear. And for the Y is something is unknown. You want to project this. You project a coordinate. You can also define some similarity in the low dimension, but actually they are different. This Gaussian, this T distribution, that's why it got the name T distributed, because it's proven to be better than Gaussian to solve the overcrowding issue. And then you define distance between the similarity in high dimensional space, low dimensional space. You want to minimize that. You can use some optimization task. So anyway, the basic idea of t-SNE is to preserve some nonlinear data similarity. So you can understand that way about how t-SNE works. So after t-SNE, anyway, you get a high dimensional data projected in 2-dimensional. Sometimes that is most frequently used 2-dimensional space to visualize, for example, the data like that, right? So of course, in single cell analysis, there are some comments about how to use t-SNE better. For example, there's some observation when you apply t-SNE. For example, t-SNE is good at revealing the local structure. Of course, it's defined based on the local data similarity, but it's not very good to review the global structure or global structures here. For example, just here, for the data after t-SNE, it's very common to see the cluster structures of the data because that is how actually the T distribution works. But anyway, after we have this, the same cells in the same cluster, for example, here, they mean something. That means they are similar. But for example, you cannot say something like, for example, these two clusters, they are more far away, these are closer, maybe means, okay, if you want to development, first go this and then go that way. That is something t-SNE is not able to do. So this is something called more global structure. Actually, people have shown that it's not very good. By default, t-SNE cannot do that. So there's some suggestion to improve. For example, you can use PCA as the initialization. Then you run the t-SNE, because actually this step is if you know more about optimization, that is called a non-convex problem. So actually there are multiple global minimum, local minimum. So actually you can provide the initial guess of PCA and then run the t-SNE algorithm, or you can fine tuning some parameters. So you need to know more things about the parameters. There are many parameters in the t-SNE. You want to do that, so maybe you can get a better result. So this is what this paper will just talk about. If you're interested, you can see more about that paper. But another thing is that actually another popular method is called the UMAP. Actually, UMAP is very similar to t-SNE, but actually its main goal is just like, to provide, first is to speed up t-SNE. t-SNE is a little bit slow. Second, I think, at least what this Nature Biotechnology paper says is that they claim t-SNE can improve the visualization and better revealing the global structure reset. So maybe in UMAP dimension reduction, the far away clusters might show they are more dissimilar than in the t-SNE. So actually here, just brief overview, what is the difference? Because the overall workflow is very similar. The only difference is, of course, how to define similarity is different. Another is the different ways to define connection. Here in t-SNE, they use something called the nearest neighbor graph to connect the points instead of just using Gaussian kernel. But we'll go back to this point later. And of course, they use different objective functions. They use different algorithm to solve this why is faster, and they use different initial point for optimization, something called Laplacian eigenmap instead of PCA or other things for initialization. But anyway, the result is something like at least in this paper, it claims it can get some more reasonable visualization of the global structure of the data. But right after this paper, there's also another paper. They claim actually because this paper claim UMAP improves t-SNE, but actually this paper just tell, okay, there are many different things here considered, but actually what is the most essential one is this last step. This is, at the first glance, this is least important, but actually that is the most important. After their analysis, they claim that is important to preserve the global structures. So actually that it was once informative initialization is used, the two algorithms appear to preserve the global structure similar well. So if you're interested, you can see more points of that. But here, I think the take-home message, because actually they are very easy to implement in the standard workflow or package. But something maybe if you want to do a more reasonable explanation of the data, you should focus more, for example, on the initialization or different hyperparameters, or become more conservative about explain the global structure of this dimensionality results. So I think this paper is also very interesting. It provided theoretical understanding about the difference between the t-SNE or UMAP or other different dimensionality methods. So actually, by what they do is actually there's one hyperparameter in t-SNE, and by adjusting the parameter, they can show actually that parameter control the attraction force and repulsion force in this dimensional reduction. For example, if the attraction is very strong, you tend to visualize the data as something very, very continuous. But if the repulsion is very strong, you tend to form different clusters. So no, they're from the same data set. That's why I say you need to be very cautious about how to explain. They're the same data set, but they all use, for example, here, for the panel, they have the row. They all use the t-SNE, but with different hyperparameter. They have totally different result. So you understand it's a development process. For example, this is very continuous, but it's very discrete. But actually that tells nothing. But I think a good thing here is they just somehow embed the other visualization algorithm here. So actually after their analysis, they show, okay, UMAP is something between the most discrete and the most continuous. Again, this force layout is something here, and the most continuous one is something called the eigenmaps. So actually, they can explain why, for example, Laplacian eigenmap can provide some good global structure, because actually, I think it's not hard to understand because the attraction in that dimensional direction is very strong. So attraction strong means maybe the global relationship is better preserved because they're more connected, some kind of, but if the repulsion is very strong, maybe only the local connection can be preserved, but the far away connections is not preserved. So I think it's a good understanding of that, probably a good understanding of how to understand the difference between algorithms. So of course, there are also some practical tips that the whole website is here, the whole link. But I think maybe the most important thing is, the hyperparameter really matter. So you run something by default, but actually suggested you understand different, at least explore the different hyperparameter in the analysis. Maybe sometimes you see consistency is very good, but if it's not consistent, you should be very cautious about how to explain that. And also here's something like the difference between clusters might not mean anything. They can mean something. For example, when the global structure is preserved, can mean something, but you still need to be very cautious about few cell clusters are very apart. It does not necessarily mean, okay, they have no connection. So that's another thing you need to keep in mind. And also the fifth thing I think is just you might need you might need more than one plot. For example, maybe in the supplementary, you want to insert different parameters to show, okay, that is a quite robust result. So next, I will just briefly introduce, because at least one thing that people claim UMAP is better than Disney is that, or one different ways at least, is how they define how to define the connectivity or similarity between cells. They use something called graph. Actually, that is the starting point from the graph-based methods. So actually, the basic idea is for different cells, how can calculate the graph. So it's just, you can just imagine each cell is one node, and for the connection for the edge, you just give some threshold. If they're close enough, you just link an edge. So that then it's naturally form a graph. So of course there are different ways to do that. Something called KNN graph is just, for example, we connect each data with with k nearest neighbor. So you connect that way. That's called KNN. There's also something called epsilon NN. It's just you set up threshold within that range. I will connect other points, or otherwise I won't connect. So anyway, it will form a graph of all the cells. So actually, the distance of a graph can be calculated use high dimensional vector, or just use PC, as we said. We use 50 PC, and then based on the distance between PC, we calculate the KN graph. So anyway, something good about KN, it's a graph. It's no longer something like coordinates. It's just about connectiveness. Of course, you can also have the weight about the graph. So there's another direction of dimensionality reduction, is that we just have a graph. We show the nodes and the connections. And how can we just visualize that graph? This is another natural way, clever way to visualize the data. So actually, this is something called a force directed layout of the graphs. What they do is actually, based on the graph, they can define, this is something like a physical model. They define some force, just like attraction force, repulsion force, and just like the system, to take the balance. And in the end, it will just give you, in the two-dimensional space, it will give you, for example, a very balanced layout. So that is the basic idea of the force layout. That is another popular choice for you. Actually, this consider more some kind of more global for that methods instead of Disney. So you can just embed the whole graph into the two-dimensional space. And here's a method that maybe I personally like to use this, or because I think is more informative, or something types is more robust, is something called Peijie. It's just like a graph abstraction of the single cells. It's something just like, for UMAP, you only show something like that. But for Peijie, it just show you a graph. Actually depends on how you implement the algorithm, or actually they have a package, depends how you choose the parameters. Actually, you can make the relative position of the cells quite similar to UMAP, but different thing here is you have the links. They tell you more about links. So actually these links is something called connectivity, actually tell more about similarity, because I think one drawback of UMAP is sometimes, when the global structure is now well preserved, the link might be not so meaningful, or it's not apparent. For example, this one and this one, can we say there's a link? But actually here, just by Peijie, they have some algorithm to just directly, not only about their relative position, but also directly show the link of different clusters. If there are many links from the clusters, means maybe they're more they're more similar or closely connected. So that's why I think graph abstraction provides something called a global topology of the data. It's always the trade-off between local similarity or the global structure of the data. So actually, graph is also important, because after a graph, it can also depend on PCA. Anyway, you construct a graph. Actually, for the downstream analysis, for example, the most popular algorithm for clustering is actually based on graph. So actually, you have a graph, then you find the communities in that graph. That is how, for example, how Lowen or Leiden, for example, in GenPALS, they work. They build a cell graph first, and they cut into different groups. So then it formulates the cluster of the data. So actually, in principle, I would just want to make one remark here. Dimensionality reduction and clustering, in principle, they are independent procedures. So actually, one goal is, as we said, is project. Another goal is to put the cell into different groups. But actually, if the most popular, popular pipeline of clustering use graph. And as we also said, UMAP also use graph. If these two graphs somehow is similar, you can, when you plot, you plot, project back the result of UMAP, there's some kind more similar. But in principle, they are decoupled. So for example, in single cell analysis, maybe you might think there's some claims say, okay, I do this clustering and this clustering is based on t-SNE or based on UMAP. So maybe that is a wrong claim because actually they're independent, because very likely the clustering is from cut graph. And maybe some people, okay, UMAP is better for clustering. That's also a very, very sound claim, because actually you can say they're consistent because it's based on the same graph, but they use different ways to visualize that. So that's only one comment. So actually that is the first paper to introduce this idea of community detection and do the clustering. Another thing in the in the single cell analysis. So I think I will make one final remark on dimensionality reduction. We have talked about so much. This is something about critical reflections. I think this paper is it's actually preprint, published. It's not published, released not very long ago, and this is a very heated debate or discussion about that, right? Whether so actually this is what I quote from the abstract of that draft. It says, okay, they find extensive distortions on both the global and local properties of the t-SNE and UMAP. Actually, that is the main claim. So actually, but I think after we are actually mentally prepared to accept that, right, with the previous slides, because we already discussed, actually, there are many drawbacks about t-SNE or UMAP, about their limitations. So it's not surprised at this point to see this claim. So actually what they do is actually they invent something called Picasso embedding. It's based on autoencoder deep learning to represent relations. They think, okay, this is something like arbitrary, because for example, they can embed the things into a world map, this elephant, and they say, okay, even they do, this is something called spacious art. After they do this, they make some analysis, and based on their metric, they think, okay, even if they do it this way, this is not worse than t-SNE UMAP under certain metric to quantify global or local properties. That's why they say, okay, if you think this is spacious art, then how about t-SNE UMAP? Is that really, really scientific? It's not saying, I think in the main take-home message, not we just abort, or we don't use t-SNE UMAP anymore. It's actually, I think they are still very important, still informative, but just has to be more cautious, or just to have a better understanding of all this whole process and use them wisely in the research. So this paper also talks about something, if you're interested, you can see something called semi-supervised vision. Maybe it's because, this fully unsupervised of dimension reduction means you don't rely on other prior knowledge about biology, but if you incorporate more biology information, maybe you can get more reasonable result. That's why it's called semi-supervised, and also do latent space like deep learning way. So maybe I think if we have chance later, we can talk more about deep learning tasks. So yes, I think that's what I want to talk about today, about the basics about dimensionality reduction. And thank you so much. And maybe just see if you have any questions.
With the Ponga analysis
Mm-hmm. Yeah, this one. Yeah. Yes. You mean how is that relevant to biology, right? Or yes. So maybe just from methodology perspective, it's very easy to explain. Because for example, anyway, we have a graph like that, and for some clusters there are more links inter-cluster links. So that's why maybe in the abstractive graph you have a link, or the link is is stronger link. It just means, okay, maybe this cluster have a larger similarity or relevance, but actually that's purely from data, or what we say is unsupervised. So yes, biological how to interpret, I think it's really hard question, but they have improved that for Peijie. For example, later they have something based on RNA velocity. If you know that, they have some directions to give some meaning, biological meaning, like this is a transition, the direction can tell us the transition or directions and the speed of transition, use that graph. But yes, I think especially for this one, it's still hard to say, okay, biological, what it means, still dependent on your, on your data and the concrete process you want to analyze. Yeah, in general, it's just like a similarity or the global structure of connectiveness about clusters.
And how close, we talked about Euclidean distance before, but how close the clusters are, is that not only the cluster?
Yes, so this close, Euclidean distance is apparently only one choice, but actually, in Peijie, some people like you use cosine metric, cosine distance, or other distance. Yes, from my side, I think there's hardly there's any theoretical guarantee. They're all empirical. It's just like try different ways, explore a little bit. Maybe for this this is good. For other this that is good. So yeah, that's my experience. But yeah, I'm not sure whether it's unified theoretical framework to explain that. Yeah. Yeah, so matrix is actually something, for example, PCA can also be interpreted as a matrix decomposition with very little constraint. But maybe you heard other things like, non-negative NMF, non-negative matrix factorization. They have imposed stronger constraints on the matrix. So they can also have some, for example, the biology interpretations of that. So actually NMF or matrix decomposition, which I think the advantage of that is, I think they're similar to PCA. It's very easy to interpret, at least easier than t-SNE or UMAP. It's very hard to interpret biologically, but for factorization, you have different metrics. Actually, you can display because actually something can be recovered into the original gene space. So yes, but sometimes, why it's not so popular, maybe sometimes not so powerful. I think it's just like, we get a trade-off between whether we want to interpret that or its actual performance. Yes. So yes, I think it's a very good idea. It's also, some people like to use that, the MF or other metrics factorization.
I guess I was recently, it's like, it's kind of arbitrary with the field sometimes. It's just like, and then it's like visualization.
Yes.
Like
Yeah.
then I guess they're proposing to use like what's happening there.
Mhm.
Data.
Yes.
Take a look at, but
Yeah.
that's thought about at all. Being like,
Mhm.
oh, I don't know how to describe that, but yeah, I could have that.
Mhm. Yes, yes. Yeah, maybe we can communicate later. Yeah, we see the papers. Yes, I think interesting direction. Yeah. So, okay.
Function, sorry. t-SNE.
Mhm.
I saw that it is neutral information based, which is cool. It's called the wide divergence.
Mhm. Yes.
Why that, not like F divergence or something else?
Yes, so actually, that is used in KL divergence is used in t-SNE, but not in UMAP. UMAP, they just replace that with something called cross entropy, binary cross entropy. So this page is, sorry, is here. Yeah, sorry. You see the second, they use something called cross entropy instead of KL divergence. They also claim there's a one improvement over t-SNE about a different objective function. Yes. It's also other use optimal transport or what's a space. Yeah, the more fancy space. Yeah. Other questions?
Okay. If there are no
Okay.
more questions,